Thanks, Jackie. Just want to start by acknowledging the traditional owners on the land of which EnviroDNA operates, which is the Wurundjeri Woiwurrung people of the Kulin Nation, and pay my respects to their elders past and present. I'll also mention that I'm coming to you from Gunai Kurnai land, which is out in eastern Victoria. I want to acknowledge the traditional custodians on the land in which everyone is joining us from and beyond, and I extend that respect to all Aboriginal and Torres Strait Islander peoples here today, recognising their continuing connection to land, waters and culture, and acknowledge that sovereignty was never ceded. Thanks, Luke. So a bit, for those that aren't familiar with EnviroDNA, I'll just give you a really quick rundown of who we are. So we're based in Melbourne. We have purpose-built labs in Brunswick, purpose-built eDNA labs in Brunswick. We've been operating now for about 10 years, and it's been really interesting and exciting to be part of the growth of eDNA in Australia over that time. Where things are now is certainly much different to when we started 10 years ago and even 5 years ago. Importantly, even though we are primarily a laboratory-based organisation, in addition to our wonderful molecular team that does all the hard work in the labs, we also have a great team of data scientists and also field ecologists to help you sort of design projects end to end and help interpret the data that comes out as well. Over the time, we've worked across a range of different clients and industries, from government agencies, resource industry, not-for-profits, and one of the most exciting things about eDNA is its broad applicability across all those different industries and habitats and species. Makes it a really useful tool. So we've done over 100,000 samples, Jackie. I reckon that's a bit of an underestimate. I think the lab might argue more than that. Certainly feels like a lot more, but there's a lot of samples that have come through the lab over the years, and that just continues to grow. And then we've also developed and validated over 150 qPCR assays for various species, everything from platypus and fish species to decapods. Whole range of assays there. So I guess we have a really broad range of experience and capabilities. And one of our goals as an organisation is to try and share this with yourselves as the end users, the people that want to apply eDNA in the field, to help everyone get better outcomes. And, you know, that real end-to-end project capacity from survey design, lab analysis, assay selection, and then interpretation of the data at the other end. So EnviroScore is just a part of this and one of the ways that we're trying to do this. Next. So eDNA as a survey methodology has some really great advantages. It's non-invasive, it can be highly sensitive, detects a range of species. This is really well established, particularly in aquatic systems, but increasingly we're seeing this methodology being applied in terrestrial systems and marine and groundwater systems. And like all methodologies, there are avenues for uncertainty. So, and that's inherent in all survey methodologies. So, you know, I've worked with wildlife for years now, and a lot of our live trapping surveys can have low sensitivity. So you end up with an increased risk of false positives. If you're doing acoustic surveys, they're biased towards those louder, more raucous species, and some of the animals that don't call so much don't get picked up. Visual surveys can struggle to pick up cryptic species. So that uncertainty exists in every survey methodology. And that doesn't mean that they're not useful. It's important that we understand that uncertainty, recognize that it exists, try to understand where it comes from so that we can identify it and incorporate that into how we interpret the data at the other end. And this is really what EnviroScore is trying to get at. Not that it covers all avenues of uncertainty, but it's a small part of the puzzle that we can increase confidence in the outputs at the end of any eDNA analysis. And this really came about because, particularly myself and the other project managers, when we talk to clients, one of the most common questions we get is when people are inquiring about projects is, can you detect this species using eDNA? Or do you have a reference sequence for this species? And it sounds like a really simple question. And the answer can be a simple yes or no, but behind that is a lot of complexity, and it relies on a number of different avenues of evidence that need to be assessed and considered. And typically we do that in the background, but EnviroScore is a way to, I guess, standardize that and make it inherent in how we discuss this with people. And it gives us a nice way to structure it and provide that output to clients. To take a quick step back, because I'm not sure where everyone is in terms of their eDNA experience, but I'm just going to give a quick 10-second overview of a typical sort of meta barcoding analysis. So at stage one, what we start with is we collect samples from the environment, and they can be typically water samples, but they can also be scats or soil or air samples. When they come into the lab, the first step for any type of analysis is the DNA gets extracted and purified. And within that sample, there is fragments of DNA from a range of different organisms, everything from bacteria and fungi, hopefully to the target species or target group of species that you're after as well. We then apply amplicon-specific primers to target the group of taxa that you're after. That then goes through a sequencing process. So we get lines and lines, we get millions of lines of sequencing code out of these samples. And at that stage, we don't know who the DNA belongs to. So the critical step there is having a reference library of known sequences so that we can look at the sequences that were derived from samples, compare them to the reference library, and then be able to put a whole list of species out or taxa out from those samples. So that reference library is a really critical step. And it's also an avenue of uncertainty. And that's what we're going to talk a little bit about today, because we don't have reference sequences for all species. We don't have reference sequences for all different subpopulations for species. So there are gaps and uncertainty that exist there. So coming back to our question, can we detect this species using eDNA? And there's a couple of different, I guess, answers to that question. First of all is, can we find that DNA in the environment? And that's really related to survey design. So how many sites you're doing, where are those sites located? What is the sampling method you're using? Of course, sampling effort is important. So volumes of water or replication of samples, or how long you're leaving devices out for. And then, of course, there is the species ecology. So abundance, how many are there? How likely are we to detect them? What's their likely habitat? What's their behavior and activity patterns? And that's a really important part of the puzzle. That's something that we're not going to talk about today. We're going to talk more about the lab side of it. But part of it is, so one of the questions that we often get asked is, how many samples do we need? And it is a question that is difficult to answer in a very straightforward way. But we've done an analysis across a range of different species, a sensitivity analysis where we can look at how many samples we're taking and what's the probability of detection. And across a range of taxa, we're seeing pretty consistent results. So that 2 to 3 samples per site generally gives us pretty high detection rates across a range of species. And that's often a starting point that we use for that question that comes up. Of course, we can also run pilot studies for your particular species or group of species and do this type of analysis for your project. The other side of that question is, once those samples have been taken and they're in the lab, does the analysis have the capacity to identify the species of interest if that DNA is present? And of course, integral to that is, do we have a reference sequence available? If we don't have a reference sequence, then there's no way that we can identify that to a species level. But there's a lot of other stuff that comes into it. So what's the assay that we're using? So different assays can target different gene regions, and that will influence the what reference sequence availability is there. What similar species occur in the area? So genetically similar species. So typically species within the same genus, and do we have reference? Do we have the ability to differentiate your species of interest from those other species that occur at the short gene region that we're using? And that's really the area that EnviroScore is trying to provide a bit more clarity around, and also identify maybe where there's gaps in there and what's our way forward to provide more confidence to detect your species. Final thing I want to do is just kind of, I guess as myself as an ecologist, I sometimes get a little bit overwhelmed by genetic terminology and analysis. And I just wanted to bring it back to eDNA metabarcoding analysis is really similar to other techniques that you're probably more familiar with. So things like camera trapping or acoustic monitoring, they're very similar to eDNA metabarcoding. So I'm going to trial a bit of analogy for you. Hopefully it will bring things, make a little bit more sense to you and sort of demystify some of the, I guess, terminology around metabarcoding. So the first stage is always going to be site selection. So where are you putting your camera out? What type of habitat are you targeting? How are you orientating your camera? That's the same as, where are you taking your environmental sample? So your water sample or your scats or swabs. And it really should be related to your species ecology and your survey question. It's something that often gets mistaken around thinking about your species habitat, preferred habitat, its movement behavior, and also its migration patterns as well. Second is what's the type of equipment you're using. So certain cameras are going to be better for some types of species versus others. Are you using infrared cameras to target nocturnal species or not? Or taking a step further, cameras can be really good for, say, terrestrial mammals, but you might want to pair it with an acoustic monitor to look for, say, frogs or bats. So your equipment type is directed towards or more favorable towards certain types of species. And that's similar with your assay selection. There's a range of different assays that we can use. They're going to target particular taxonomic groups, and we can stack those assays together on those samples to provide broader analysis or more confidence of your group of interest. And then, of course, no one is looking at hours and hours or thousands and thousands of images anymore. No one's manually going through them and checking them. Most of them are going to be sticks waving in the background. Of course, now it's all automated, either using a software or an AI, and that automation needs to be trained. So your method of analysis isn't going to be able to identify your species of interest if it doesn't know what it looks like. So the more images that you can provide to that, the more trained it's going to be, and the better confidence you've got of detecting your species. So if you can provide it with a heap of different images from different angles and different lighting, different color morphs if they are of your species, that gives your automation more strength. And that's the same as the reference library for your genetic signals. The more reference sequences we got for a species, the more representative of the population we can understand, and we can see whether there's variation across that population as well. And then I guess the final part of it is how well can we actually identify that species? So is it morphologically distinct? So you can identify a giraffe much easier than you can, say, a small rodent. What other species occur in the area that look similar? So if you've got 3 or 4 different rodents in an area, can you distinguish between a bush rat and your feral black rat? How well is the image quality? Clear images are much better. If you've got sort of blurry ones, or you've just got a leg that's disappearing out the edge of an image, makes it much harder to identify. And this is really analogous to the phylogenetic signal of our species here using an eDNA analysis. So how genetically distinct are they? How well can we differentiate it from similar species that occur in the area? And I guess those last sort of 4 aspects there is really what EnviroScore is designed to address. It looks into all of these different aspects, uses multiple different data sources to really bring all that together and understand how well we can identify species in that laboratory analysis. That's enough from me. I'm going to hand over to Luke, who will delve into more the technical side of it and the details, and I might see if I can answer some of the Q&A that's coming through. So thank you, everyone.
Thanks, Josh, and hello, everyone. Thanks for joining us. I think it's fair to say confidence in eDNA is a topic of pretty high interest to people who are already using eDNA or those thinking about using it. It's a testament, I think, to a slightly maturing field, but it is, as Josh mentioned, a really broad term. And so I expect that some of the people here are already disappointed in the scope of which we're operating, but we will continue with that sort of modest scope in mind, and we will be continuing obviously to work on all aspects of confidence broadly conceived, as I'll touch on later. So before we get into some details on what this score is and what it's doing, a quick refocus on scope. We're looking essentially at two phases here. One, what comes before you do anything, that is the planning phase. How do we know that the tools, the molecular tools that we're using are fit for purpose? And then also increasingly at the detection stage and the interpretation. I will make a quick diversion into the methods that are both sort of in scope for EnviroScore. We're obviously using sort of targeted methods and broader methods. I guess another implicit frame here in this discussion is a lot of this work is geared towards threatened species. So, sometimes communities of species, often single species. So we're considering both metabarcoding and qPCR assays, which typically sort of map onto detection platforms, but don't necessarily have to do that. Really, the main distinction is where do you address the uncertainty and how do you resolve it? So you can do targeted work on sequencing platforms just fine, and increasingly we're doing that for the added confidence that it gets you. So what these methods demand is, of course, in a perfect world, complete reference data for all species that you care about in a particular place and time, and also all of the species that you may care a bit less about, but need to know something about in order to resolve what you do care about. And so for designing a targeted assay, that all comes up front in the sense that you assemble a set of data in order to guarantee specificity for your target group. In a perfect world. And so you're essentially designing out some of the specificity risk at the initial assay stage, whereas that comes, in a more complex metabarcoding context, you can't do that for everything, of course, and we're still living in a world of, imperfect databases. There's also the fact that even in a world of perfect databases, you will never have perfect discrimination. And that's for multiple reasons, some of which we'll get into that are both sort of technical, biological, and human. The detection evidence for these platforms tends to differ. As I said, if you're using qPCR, you'll often be working with sort of binary detection, non-detections, though that, of course, is you get quantitative DNA counts out the other ends. And whereas for metabarcoding, you can be looking at binary or relative read proportions. And so, there are different ways in which you handle uncertainty. I would say this reference data is probably the most banged on about thing when it comes to eDNA uncertainty. So I won't labor the point here, but it is obviously a fundamental thing, as Josh mentioned. Indirect methods rely on reference databases to do interpretation. And so we need to have information both on target species, all close relatives that could be expected to occur in a particular area. So that is implicitly a spatial question. And not just are there any sequences, but you have to look at the quality and quantity of those sequences. So do you have enough to reliably assess genetic differentiation? And what enough means varies with barcode that evolves at different rates. It varies with taxonomic scope. Different taxonomic groups evolve at different rates. And then, of course, does the genetic signal that we see map cleanly onto taxonomy, which is, like most things, when you scratch the surface, and an attempt by humans to sort of map discrete things onto a moving target. So a continuous quantitative sort of signal, which is evolution. So obviously the state of reference databases is getting pretty good. It's getting better, very unevenly, of course. We're pretty lucky down here in Australia, as I'll touch on the next slide. But this is a plot of all vertebrates, mitochondrial barcode sequences that are going into one of the major public repositories, NCBI, out of the US. And it's clear that things are improving rapidly. But improvement itself is an issue in that reference databases are now changing quite rapidly. New sequences are added, taxonomy is updated or revised, amplicon coverage can be extended or assays improved. And so really results and a scoping of a project really need to be refreshed, repeatable, and tied to a specific database state. These are the numbers in terms of proportions of native Australian vertebrates through time. And as I said, we have, there are comparable global efforts, both, small and large, that are boosting coverage. But in Australia, we're lucky to have some major programs coming out of Australia's National Science Agency, CSIRO. Their biodiversity library has basically given a huge jump to marine taxa. This is, you can see jumps there for fish and sharks and rays. And there are new data coming out expected in the next few months for terrestrial species, and then other campaigns for far more challenging things like marine invertebrates, fungi, plants, and macroalgae. But that's not enough, right? Because A, this is not enough data to reliably assess within population differentiation and variability in some cases, and also its phylogenetic signal at mitochondrial barcodes or commonly used barcodes is not always enough. And so, unfortunately, we still have to fall back in some cases to more targeted assays that are more sensitive and can sort of employ more rapidly evolving barcodes that are less suitable for metabarcoding. So let's have a look at some of the source of information that we're looking at in EnviroScore. The aim is to quantify taxonomic resolution at the project scoping stage initially, identifying where existing methods can discriminate the target, if there are data gaps, whether existing qPCR or metabarcoding is appropriate or neither, whether new assays might be able to help that, or whether this is just a hopeless case for eDNA and it's genetically complex or taxonomically disputed. What does it do? We look at reference sequences, of course, as I mentioned. We look at this in a spatial context to use occurrence data or expert curation, so species distribution models from federal government where they're available, to assess basically what is the risk for a particular project and increasingly at a detection level. We look at both public, essentially any form of occurrence data. At the moment, it's mainly ALA, could be and GBIF, but it could be other forms such as passive acoustics, private or public data. We look at phylogenetic signal across validated assays, metabarcoding and qPCR, and we look at the validation state of those qPCR assays. It's not, of course, enough to just have an assay. Assays vary in their quality and level of validation from in silico analysis through lab technical validation, through validation in the fields, and then ultimately through modeling against existing methods and potentially environmental covariates to arrive at the gold standard of a finely calibrated method with estimated false positive and false negative rates against a particular sampling method and design. Not always achievable for all taxa, but as Josh mentioned, we're essentially working through as many case studies as we can. We're not really at the stage, I don't think anyone in the field is at the stage where you can generalize from these case studies to any taxon, but it is a task for the field to get enough of these gold standard case studies where we can start to make more finely scaled principled sampling designs in a repeatable way that take into account eDNA ecology, species ecology, hydrology, all these other things. So essentially, I mean, this is the EnviroScore is not a linear pathway. It's essentially a thresholded set of logical gates where, again, we're converting continuous things into a simpler sort of traffic light system. So trying to bring some simplicity to what is obviously a fairly complex thing. And so the minimum requirement for a green would be the presence of multiple reference sequences for a target species and all closely related species. And so in simple cases like platypus or lungfish, that's a very low bar. And then a strong history of detection for some assay, whether that be qPCR or by metabarcoding using assays that show a clear barcoding gap between target and non-target species. I guess the main thing that it's really resolving in the field now is incomplete databases, which, as I said, is to some extent a temporary problem, but also differential phylogenetic signal across barcodes and assays, which is not a temporary problem. And also flexibly integrating others of sources of support for species distributions and allowing us to sort of QC that data. I mean, we're not taxonomic experts as much as we have some, some depth in some areas. So we rely on public data, we rely on experts. The best we can do is sort of be transparently curating that data, which we know is necessary. There are errors in reference databases, there are errors in occurrence databases. And we need methods to sort of flexibly and efficiently screen those and be transparent about what we're using or what we might not be and why. So I think most people here, if they've thought about eDNA, probably know some of the major sources of uncertainty here that, taxonomic issues and how something like EnviroScore might help. I'll run through a couple of really simple cases, but I don't want to, again, labor the point around the complexity here. So essentially, in a simple case where we have a monotypic genus like platypus and we have good reference databases, we would call that a green. That's a high confidence, and there are not many cases that are that simple. In a simpler case here, one assay, we've got a target and a congener. We have good adequate representation for those and a good barcoding gap. Again, that's fine, easy. Things get more complicated quick. Here is a case where you might have multiple barcodes and there is overlap in the phylogenetic signal for this particular contrast. And so that is a case where you may have confidence, you may not, depending on perhaps where those sequences came from, where your detections are relative to the known species ranges for these two taxa, and also your confidence in whether those distributions are actually accurate. As we're seeing with eDNA now, we're seeing range expansions beyond what is known from traditional data in many cases. So we have to be careful about overly relying on historical data. It's a balance because we don't want to be just reporting what people already know. We want to be transparent about what the data says against current reference databases, but also transparent about what our best prediction is given XYZ. Things get more complicated in the case of data gaps, right? So obviously, if you have gaps for a particular taxon, you can get misassignment. Here, we're taking, again, sort of a target congener framework, but this is a general problem, regardless of what you consider a target and a congener that may be of less interest. And clearly, in the case where you have no sequences, you have no ability to resolve that. And this is a single amplicon case with just 2 species. So things get much more complicated much more quickly when you're considering many species with variable data gaps and you're considering multiple amplicons, which the field now is widely using, and for good reason. But it is simply not the case that multiple amplicons provide universally better data. They provide better data when it is interpreted better. So we're going to look through a couple of case studies here. And of course, happy to answer questions about others later on. And so we'll have to stop sharing and switch windows. Which I will do seamlessly. All right, I'm going to assume everyone's seeing that. And Josh or Jackie is going to confirm that for me.
Yep.
All right.
Good.
So let's look at one of our favorite turtles, Alsair. So this is just a demo. We've put a few species in here. This is not by any means all data. We've started with all threatened species under federal and state jurisdictions, but of course this can work for any taxon, which is the direction we're going in. And so southern snapping turtle, this is a case where we actually have a ton of data, both from metabarcoding, qPCR, and traditional methods, netting and snorkeling, the like traditional surveys. So in this case, we've got a clear green. We've got an abundance of sequences that are public, and that can be any sequences. I mean, sometimes any sequences is better than none, at least tells you that there is DNA somewhere and someone studied it and there's a chance of getting tissue if you need to design an assay. Of those, it tells you which ones are most suitable for metabarcoding. So in this case, we've got sequences for 12S and 16S, common metabarcoding targets. Tells us that there are two potentially co-occurring congeners, or sometimes confamilials, if we need to expand the taxonomic scope. But in those cases, we have both sequences and we can assess the barcoding gap. And so in this case, Alcea is monophyletic. It's got a good barcoding gap. There is no variation within it for the assays that we've selected. And we have a strong detection history, both from metabarcoding and qPCR, although in this case, we're sort of gating 10 field detections as fairly moderate. The data for this is continually going in. We don't actually have all of our data in there yet because it goes back a long way. You can also look through sort of explore the public data. Again, this is a fairly simple case in that we can bring in, SDMs for multiple species. In this case, we have it both for Alcea albigula and Irwini, which is up here in the north. There's really no confusion risk here. If you bring in the ALA data, with the exception, you can already see there's an outlier here. This almost certainly a wrong occurrence for Alcea albigula up in the northern snapping turtle range. And so again, as you would expect, I'm taking you through a fairly nice, simple example. This is very much a green. And then we can bring in, of course, our historical eDNA detections, and that is sort of a reinforcing the evidence for scores as we go through time. We can look at the spatial signal of all of the amplicons that are in scope, and we can look at historical detections. I won't go through all of that just yet. We can also start to look at broader trends around, which assays and which barcodes work better for particular groups, where the gaps are. Is it mostly due to reference sequences? Is it mostly due to phylogenetic signal? Is it, are there essentially just no good assays for that particular taxon yet? Glassfish. Let's have a look at another one which is a little less clean. And so the reasons for this being amber are essentially that we have incomplete information for potentially co-occurring congeners. In this case, it's still fairly optimistic in that we have pretty good reference data for most of them. We're missing one. Strong phylogenetic signal and really strong detection history for metabarcoding, but in this case, no qPCR. So I hope you can see essentially that, any of these things, if we're taking a conservative approach and we're demanding essentially that we do have 100% coverage for all co-occurring congeners expected. Obviously the corollary of that is this is a spatial problem. This is an assessment at this stage here of all of Australia, essentially. And we can take this anywhere in the world, but this is not a spatial signal yet. What we want to do, and are starting to do, is to then take this to the detection level. Where we can now assess for every individual detection, what is the confusion risk there? Because obviously species ranges vary. You might have perfect confidence at one end of a species range, intermediate confidence where it abuts another species, and maybe no confidence where there are data gaps. So data gaps vary in space, and so do detections, and therefore so does confidence. So where we're moving is to essentially have a transparent score on, again, what the algorithm says is the best match given today's reference databases. What is our prediction of the most likely match given an assessment of gaps? And then also essentially a version control for all data so that we are able to cope with the ever-changing improvements in supporting and reference data. And we are able to tie a particular pipeline and a particular data set to a particular set of results, but flexibly update that as new information comes to light. So obviously, there's more to do. Data science cannot fix improper sampling method, design, or laboratory procedures. So we need all to be working on that as we are. Where we're also going is more data-driven aquatic survey designs, as I mentioned, where we have enough data now that we can start to make very general recommendations about power for particular places and times and taxa. And then, of course, a longer-term goal is applying these things to emerging terrestrial survey methods like air, scaled scat collection, etc., etc. So I will finish that. Hopefully, you have time for some questions, and of course, happy to answer any more offline. Thank you.
Nice one, Luke. Yeah, there's a few questions that have come through. I have added some responses, but I think it'd be good to get your take on these as well. So first of all, it is how likely is it that a species that is genetically known, i.e. in the database and present at a site, but not detected in an eDNA sample, assuming sampling methods are good? So I guess what's the chance of false negatives?
Yeah, depends on what the taxon is. And so if it's something that we have calibrated, like a small fish or a platypus or a particular frogs, and we know where you've sampled, how you've sampled, when you've sampled, we would be able to put numbers on that. There is no general answer for that question. So it comes in, unfortunately, with a bunch of sort of implicit constraints in there, which means you can't have a general answer. I'm sorry, that's very unsatisfying. If I know more information, I could probably give a better answer.
And look, I'll just add to that in aquatic systems, at least, there's a number of papers that have been put out there comparing those false negative rates with traditional methods. And typically eDNA, the sensitivity is much higher, so those false negatives are much lower. But yes, it is going to be a species by species case specific and also environment specific as well.
Yeah. All we can do is work through case studies. We're doing this for freshwater turtles at the moment, again, where I mentioned we have quite a lot of data. Obviously, it takes time to get that into sort of regulatory approved methods. But if we can show that there's a clear improvement over traditional methods and we can show the effects of sampling design, that's essentially how we can move forward. It's a different question when you're doing broader metabarcoding. There's very different questions and different power dynamics.
Another one here in terms of contamination. In terms of building your scoring system, one of the key challenges I had to deal with is contamination from human to many others, which can affect the quality and your expectation. Yep. Any advice?
Yes, I guess. I mean, humans, are everywhere, and that can depress sensitivity. If you're using general methods, it can only depress sensitivity. And the same goes for, perhaps less uninteresting things like carp in freshwater environments. That can be handled to some extent in the lab by, again, the answer depends a little bit. If you're doing targeted things, there is, very specific things you can do. You can exclude those taxa by, targeted or somewhat more general means. In the case of metabarcoding, it is much harder. You can't just block out, 10 taxa that you don't care about. You can apply blockers to humans one at a time, or you can apply blockers to carp. You can maybe apply a few blockers. I think 3 is the most we've ever done. You start to get distortions in the data, sometimes even with one blocker. So the other approach are different sequencing methods with different sensitivity. You can sequence very, very deeply. You can do more cutting edge things, but the fact is that human is everywhere. We just need to kind of deal with it by building that in. So when we consider QC, we exclude human because we know A, it's a potential laboratory contaminant, but B, it's of no interest to anyone. So our QC thresholds are built around returning data to the client, which is useful, we hope. That still doesn't guarantee that there's depressed sensitivity for particular rare taxa. That can really only be addressed by more targeted methods or better sampling.
One that just came through from Jai. Are threatened species with lower genetic diversity likely to get good data references sooner than common species and species with high genetic diversity?
I don't know. I'm not sure if I could answer that. I can tell you that the accumulation rates in databases for threatened and non-threatened native taxa are pretty damn similar. So it's clear that there's no systematic efforts that I can see in the public data. I couldn't speak for CSIRO as to how they are prioritizing taxa and whether that's their scaling effort with expected genetic diversity, which, you know, would be sensible, but is, you know, not a non-trivial exercise. So, yep, don't know.
I think most groups would want to do that, but for a lot of these threatened species, you know, some of them haven't been seen for 30 years, and so trying to capture them to get genetic reference sequences is just a bit of a challenge. What else do we have here? There was a comment. Let's see if I can find it again. So Joel mentioned, so this is very vertebrate focused. Does your score consider taxonomic uncertainty or incompleteness? E.g., do we not know if there are undescribed species in the genus, such as you see in many invertebrate groups?
That's a hard one.
Yeah.
I mean, we can look at published diversification rates across groups. You know, they're not obviously universal, as some proxy of that. They're also not, you know, universally available to assess risk. So that we have diversification rates in there, but it's not, as I said, is not as widely available as we would like as other. So we sort of view that as a it's not scored essentially in there. It's in the report. Invertebrates, very, very difficult, as you know. I mean, look, there are algorithmic methods that attempt to, you know, do this. There's, you know, evolutionary placement methods, or there are methods that take both, you know, backbone taxonomic trees as well as genetic data and try and sort of infer hidden things, or at least appropriately tailor confidence to particular branches of the tree. I think it's a very, very hard problem. And I think you probably know, as your brothers and your question, that there's just an enormous amount of uncertainty in both the quality of the genetic data and, you know, the breadth of it for invertebrates. So it would have to be sort of tackled, you know, clade by clade, really.
I've got a good one to throw at you, Luke, because this is a question that I think always comes up in these discussions. How far are we from quantification of a certain species? So not copy numbers, but number of individuals.
No general answer to that is the coward's reply. I guess this is more broadly, this is sort of an effort to get us to a place, we hope, where instead of, you know, scrutinizing single data sets and asking, you know, how much information can they tell us, and agonizing over every single, you know, read in that spreadsheet or whatever it is, a place where essentially we are a bit more probabilistic, and we're using, you know, ongoing routine monitoring where essentially you can end up with probabilities of detection and non-detection, and also tailor that to traditional data. So again, catch data where you can sort of anchor it in questions of, you know, biomass and individuals. And in some cases, you know, we do quite a bit of population genetics using related eDNA and non-eDNA methods. So using that kind of information, but it's really only going to be approachable for really key species where, you know, there's a lot of effort, whether that's conservation species. We're doing, you know, a lot of work on this with lungfish, for instance, or commercially relevant species. You can get there. You just need to collect more data, and not just eDNA data ideally, so that you can start to put together models that will more reflect biomass. But it also goes to the broader question of, you know, what are you trying to do? And again, the answer depends whether you're thinking about conservation or commercial fisheries. If you're considering the sort of genetic health of a species, there are other things you can do. I mean, you don't always need to know the actual age of things, or perhaps the absolute count of things. I understand for different regulatory methods that information is absolutely essential, but there are other things you can do around genetic health that give you both a sort of a recent and a long-term view of how that population is tracking, that, you know, it doesn't necessarily require accounts as much as that may be useful currently.
Just to quickly add to that, because I do get this question a lot, is that, I mean, essentially from my point of view, abundance is a way that we infer population health. There's different metrics that we can use in terms of distribution and area of occupancy and site occupancy that will tell you the same thing. So I'm using it a lot for my platypus surveys now. And it's actually, I find it's a better metric of population health than localized abundance. The other thing we have been doing for a variety of species is we can actually genotype individuals from scat samples now. So we've been doing this with some invasive and native species, so we can actually quantify individuals from scat collections.
Yeah, I guess I would also say on a slightly more optimistic note, I mean, the field was very cautious about this early on, as it should have been, but there's pretty good emerging evidence that, at least the rank abundance of fish biomass can correlate with metabarcoding when you do it well, right? And you do it consistently. So I don't think anyone's, well, I know some people are doing this in a fisheries setting where there's fairly small numbers of taxa and you can really hone the parameters involved with correcting bias and things like that. But most people are pretty cautious about converting what we expect to be at least a rank abundance correlation into numbers. But we know there's useful information there when you do things properly, at least for fish.
Okay, a couple of final ones. Hopefully a nice easy one for you, Luke. What do you mean by scaled scat collection?
Yeah, lots of scats. One of the favorites. The lab always likes to hear this when it goes out. Gut contents and lots of scats. That essentially means detection dogs, really, where you can do surveys and you can just basically, multiply the numbers that you can pick up. If it's important for you to look at, in terrestrial environments, they're pretty good proxy for, to see what things have been eaten. You can look at native small mammals, for instance, and birds. Or, of course, you can do genomics on the predators themselves, if that is important, whether they're native or feral. But yeah, detection dogs, that's pretty much how we scale.
Okay, a couple of ones just to finish off with. Much of the score seems to be about characteristics of data around the species, but how do you interpret the raw data? This is probably something that I tend to chat with people about around presence and like read counts into presence and absences. So if you have a green scoring species but low reads, or conversely red species with high reads in your data, are there thresholds? I think this probably doesn't relate to read so much. It's more of that data interpretation at the other end.
Yeah, I mean, we're heading in that direction, right? Where you can look at a detection level and then read counts and replication, as well as just bringing in other information, spatial information would be relevant there. I mean, if you have a species that is read for us, in a particular project area using an assay that cannot discriminate it, well, it doesn't really matter how many reads you've got. That species level assignment is suspect, and you need to be clear about that. If you have low level detections, again, I mean, this kind of goes to the point about how much information can you get from a single snapshot. I mean, we look at replication, just the workhorse of experimental design. So is that detection replicated spatially, temporally? Or from other projects, perhaps, rather than trying to, put a lot of weight over a single detection. We know it can be sensitive, but there's, nothing is going to give you more comfort than seeing that replicated by either eDNA or other methods. And we've seen that time and time again with eDNA, surprising results getting replicated when, the effort gets put into going out and surveying. That's not to say that all results are 100%. I don't think that's true either. But replication is the best friend there. And we know that recounts can vary for a whole bunch of reasons, right? So you can have some taxa just don't shed a lot. And so again, if you're looking through time, that baseline is low. It's never going to be up there with other things. If you're doing vertebrate megabarcoding, you're always going to get messy fish. More than turtles. But again, it's the relative amount and the replication that gives you confidence.
All right, one final one, and then I'll let you off the hook. If other ones that we don't get to, we'll try to respond to people after this session. I've lost it. Is a metabarcoding approach to surveying for a list of species, such as a list of threatened species that may occur at a given site, more appropriate than conducting multiple targeted qPCR for each species?
It's a necessary trade-off in some cases. Basically, it depends on your list. There's a trade-off, obviously, in how many assays you can independently run, before you use all your DNA. I mean, you're doing qPCR one species at a time, or even, a few species multiplex is a very inefficient use of a precious resource, which is, the DNA you've collected from a sample. And so again, it depends. We have done a bunch of studies. I mean, qPCR, you have to expect when it's done well, it is more sensitive. But again, the trade-off is you can do less with it. I think a better approach is to make sure that your metabarcoding panels are semi-targeted. For instance, that is a more efficient use of the data, and we know in some cases it can give you as sensitive a data as qPCR. So that is something we would like to do more of. What you don't want to do is use super broad assays for all eukaryotes, say, or sometimes even all vertebrates. If you're only interested in fish, is too broad. You're just depressing the sensitivity. So as always, the answer depends a bit on how long the list is and what your needs are. What are the consequences of a false negative, essentially? Is it catastrophic? Is it biosecurity? Or is it, environmental impact where you need to prove a negative? These are all things that are a bit contingent, I'm afraid.
Excellent. All right, we might call it there. I think we got to most questions. Anyone that we haven't got to, we'll try to follow up after this session. But I think I'll let Luke go and have